Hello, everyone. This is our interviews about what we're reading. And today we are with Matthew Smith. Hello, Matthew.
Hello.
Matthew Smith is an associate professor at University of Florida. And today he's going to tell us about his recent publication in Mycology entitled Systematic Study of Truffles in the genus Roulandiella with a description of two new species from Patagonia. So, Matthew, what can you tell us about this publication?
Well, I'd be happy to tell you a little overview about what the paper is about. First, I want to say that the first author of the paper was a master's student in our lab, Natapol Chrysit Udom Suk, and he is now completing his PhD in our lab, working on a different systematics project. But during this work, Natapol was characterizing the genus Roulandiella, and that genus are very small ascomycetes, truffle-like fruiting bodies. That's the ascocarps that they form. And they're exothecial, which means that if you look at the fruiting body, the spores are facing outward, whereas a regular truffle has a peridium on the outside. In this case, all the ascospores face outward, but they don't seem to shoot the ascospores off. So they're interesting fungi, but they're quite small, so they haven't been well studied. The genus Roulandiella so far is only known from Australasia or in places where those species have been introduced with eucalyptus, mostly eucalyptus or related Myrtaceae. Mm-hmm. So we were in these forests collecting fungi over a relatively long period of time. First started collecting these actually in 2008, and we started collecting these fungi and realized that these were fungi that were the genus was only known from Australasia, but these were clearly new species. And then the more that we looked into it, the more we found that there was an interesting historical aspect as well, because one of the species was first collected by Roland Thaxter. In 1906, he visited the far southern part of South America, so he spent time in Punta Arenas, Chile, and he collected ascocarps of this fungus, and they were pickled, and they were at the Farlow herbarium for more than 100 years, and the species was still not described. So Natapol got together a collection of Roulandiella specimens, including the described species from Australia. They're very small and they're very hard to get material. So he did a good job of tracking down the material, and then he used several loci. He was able to get sequences from the large subunit, RPB2, RPB1, and maybe one other locus. I can't remember off the top of my head. And then also ITS. So we were able to show that that these are ectomycorrhizal fungi associated with Nothofagaceae. So they're ectomycorrhizal with the roots of Nothofagaceae. And also that there are two major clades so far out of all of the ones that we've been able to sequence. There are two major clades. One is the Australasian taxa that are mostly known from Australia. And then the other are these two species that are known only from southern South America. One of the species from southern South America is incredibly common. It's everywhere. But we know it mostly from these asexual states that we find in, I can explain a little bit about those, but the other one is only known from two collections. So either it's really rare or it's in the northern part of the distribution, or maybe both, we don't really know. So that's kind of a quick synopsis, but maybe you have other questions.
Okay. Yeah, so I'm interested about these asexual spermas. If people collect them, can they identify the species based on them, and how can they use them? Because they're actually very common, no?
Yeah, they're very common. So the one species that is very widespread and very common, you find it in almost every Nothofagaceae forest that you go into. If there's moisture and the ground is wet, then on bare soil, this spore mat is everywhere. And if you look at those under the light microscope, and also you can see figures in our paper, those spores are really abundant. But one thing is we don't know whether those spores are actually conidia that are used to make new individuals and be able to establish ectomycorrhizae associations with Nothophagaceae, or if those propagules are used as spermatia for sexual outcrossing. So we really don't know the function of them. So either one of those could be possible. The ascocarps themselves are really small and gelatinous, so they seem to be very uncommon, and we don't really know how they get around because the ascospores don't seem to be shot off away from the ascocarp. One possibility is that the ascocarps are eaten by animals. They look a little bit like snail eggs or amphibian eggs, so it's possible that they're eaten by some organisms, or they may just disintegrate into the soil because those spores that are in there have very tough walls and have a lot of spore ornaments. So they may have a spore bank or something like that. But in terms of identifying them, it's really hard. You can tell that you have the right family, so you can tell that you find an anamorph in the Pisizaceae, but you can't really be sure of what the species is without sequencing DNA from those. So they're pretty easy once you learn how to collect the tissue, and then you can do DNA sequencing from the tissue, but you just have to know some tricks, basically.
Okay. And so you said that these fungi are ectomycorrhizal, right?
Yes.
So how do you know they are ectomycorrhizal? And do you think that they have important functions? Because usually when people think about ectomycorrhizal fungi, they think more about like mushrooms or in the back. And mostly they're usually very abundant, like from the Basidiomycetes. So
yes.
I think that they are very diverse in South America, for example, or do they have particular functions in these forests?
So I will say there are limited data for a lot of forests in Southern South America, but out of the work that has been done, there are a couple studies where they find them on the roots. These on the roots, they seem to be more abundant and diverse in places where there's more seasonal drought, and maybe places where you'd be more likely to have higher soil pH. So it's probably more important in the, let's say, in the eastern part of the forests in Argentina, and then probably further north as well. I suspect they become more important in those environments. But as I say, one of the species that we described, that's the really common one, Patagonica, that one is really everywhere. So we assume that they must be important. So out of the studies that have been done, the one study that I know of where one of the species was really, really abundant on the roots is in a greenhouse study of Natalia Fernandez and her co-authors from 2012. And they found that these were really abundant in their seedlings. They did a seedling bioassay with some field soil, and that field soil, for whatever reason, had a lot of spores or something, and they had very high amounts of this. They didn't call it a Rulandiella. They called it a Pisiza because that was the closest blast match based on GenBank. But it's clear from our sequences that they were finding the roots colonized by one of the Rulandiella species.
Okay.
So I think in certain conditions, they may be very abundant on roots, but we need to find out more, I guess. More studies probably to be able to answer that for sure.
So what are your plans now? Are you still working on these groups, or would you like to tell us a little bit more about further studies that you're about to do?
Sure. So we're continuing to work on systematics of Pisizalis because I would say many Pisizalis in these forests in Southern South America remain relatively understudied, and we've already submitted one paper on the genus, what used to be the genus Underwoodia, which are large Helvella-like fruiting bodies, and so we're going to be describing some new species there, and then also in the genus Amelascus as well. We have some ongoing research that's not quite ready to submit, but yeah, we're going to continue in our lab to work on especially truffle-like fungi as well from Southern South America, and in collaboration with other people as well. There's just tons of work to do. There's so many exciting things I think that we can still find from those forests. So I'm excited, and I'll be going back this year twice because I have a sabbatical. So that's really exciting to get to go back because I really enjoy it there.
Very nice. Well, thank you very much, Matthew, for your time. And we will be back soon with more interview for the South American Mycorrhizal Research Network.
And Thank you very much for having me. I appreciate it. It's a
Bye.
great opportunity to share my research. Thanks.